Good evening, everyone, and very warm welcome to each one of you. I am Sujit Nair, the co-founder and director of technical operations with Spectratix Pharma Private Limited India, and I'm so glad to see each one of you for today's session. We have around 600 participants, which is a huge number, and across the globe there have been more than 600, I guess. Now the number has crossed 1,000, and let's see how many of you actually participates in the webinar. So the webinar, as you might be knowing by now, is part of the Spectratix Knowledge Series, which is an initiative by Pharma GAN, the training division of Spectratix Pharma India, and what you say, the intention is to help students and professionals so as to stay updated with real world pharmaceutical skills and industry expectations. So that's the motto which we have started this mission. A brief introduction regarding Spectratix Pharma. It's a pharmaceutical research and training organization which has been dedicated to accelerating drug development, analytical services, and clinical research activities. And we also have great interest in bridging the gap that currently exists between the academia and industry. Through these practical training programs and dedicated research supports, we are trying to overcome this gap. And the mission, as I already stated, it's very simple, and we are trying to bring out a lot number of quality professionals, you would say industry ready experts who have real world competencies, and we want them to have that cutting edge technical knowledge also. That's why we come up with topics which are relevant, and today's topic is also very much relevant. It is CTD in regulatory affairs. It is one of the very most, or I can say the most important one in the current scenario with respect to drug filing. Regulatory affairs, as you all know, that plays a key role in our industry, and the motive has always been to keep our medicines safe, effective, compliant with respect to global standards. With the changing scenario that exists in our domestic market also, I guess these topics are becoming more and more relevant. Among all the regulatory frameworks, I feel CTD, or Common Technical Document, has become a universal language with respect to drug submissions. So whether you are working in your QA, QC, R&D, clinical research, or you are an aspiring regulatory operational personnel, understanding CTD is quite essential. As we will see in our presentation also, CTD is quite essential for our own growth also, not just from the economic or from the pharmaceutical point of view, but from a personal point of view, understanding CTD is very much important. So I am very happy and privileged to have Dr. Reshma Rajan with us today. She is an expert with respect to the subject that I was talking about, and she brings extensive experience that you will see from the presentation itself. And also, I guess it will bring a practical insight into the regulatory landscape. She is currently working as a manager in regulatory affairs at Sanzyme Biologics India. And here she spearheads the end-to-end CMC strategies for product registrations across all the regulated and ROW markets. And I guess she has a professional experience of more than 20 years now in global regulatory submissions, compliance across your biologics, pharmaceuticals, in vitro diagnostics, your medical devices. So the wide range of regulatory applications she has an expertise. So she has proven expertise with respect to your regulatory CMC, CTD submissions. All I request is that by the end of session, you should be utilizing this opportunity, bring out maximum number of questions, and I am not sure how many we will be able to take, but we will take the maximum. If we are not able to get or answer all the questions, we'll make sure that we bring the answers at least through the group itself. So we have a time limitation. We'll stick to the timeline. Okay. She is besides this professional expertise, she also has more than around 10 publications in reputed journals. I am very sure that this session will clear the concepts, build confidence, and give clarity on how CTDs will work in the real submission. So thank you so much, Dr. Reshma, and also the participants for joining on time. And let's begin the session. So over to you, Reshma, please.
Thank you, Sujit. Good evening, one and all. Let me first share my screen so that you can see the slide. Hope you can see the slide. Sujit, can you see the slide presentation?
The presentation is not clear yet. You have not started the presentation.
Started, but can you? Okay. Unable to see this?
Okay. The folder is there.
Oh, I have started sharing, but
should I share?
Wait a minute. Give me a second. Now, is it?
Not yet.
Okay. Now?
Yes.
Okay. So let's just begin. I'm going to give an introduction to the common technical document, that is CTD. This is for a beginner level. So first of all, when a manufacturing company, they manufacture a product, they'll be seeking permission to sell the product in the market. To sell the product in the market, they should have approval from the regulatory authority. For example, if I have to sell a product in India, I have to get the approval from CDSCO. DCGI is the authority who will give the permission to sell the product, commercialize the product. Same way, if it is US FDA, UK, EMRA, MHRA, EMA, like that. Different regulatory authorities will be there. So all the authorities shall give the permission for you to commercialize your product. To commercialize the product, you have to give details of your product to the regulatory authority. Then only they can go through the product details and they will check whether this product is having quality, purity, efficacy, or whatever the claims are. Is it good for the people? Then only they will approve. So for them to evaluate the product, they need the documents. So that document is our common technical document. That is the most confidential document of the product. And at the same time, this is called as dossier. Dossier filing, that is what we call for the registration of a product into a regulatory authority, dossier filing. So for a dossier filing, we need this common technical document. In today's section, these are the learning objectives. Why CTD was developed. And each CTD has different modules, or for example, different chapters. We will look into this 5. There are 5 different chapters or modules. So these 5 modules and purpose of each module and introduction to the eCTD. Okay, these are going to be the learning objective of today's session. So first question, why we need CTD or the common technical document? So previously, there is a 35 years ago, there were no harmonization of this regulations or authority rules, guidelines, nothing. Each country, they followed their own guidelines, their own format requirements, their own structure, numbering requirements, everything. Depending on the country, that varied. So after that, they formed an ICH, International Council of Harmonization of Technical Requirements for Pharmaceuticals for Human Use. So this council, ICH Council, was formed by US, EU, Japan, their regulatory authority, and the common manufacturers. They together made this ICH Council, and they thought about harmonizing the requirements for the medical products submission, pharmaceutical technical documents and their requirement. They harmonize the guidelines so that every country can follow the same thing. So pre-ICH era, each country, as I said, they had their own requirements, their own regulations. So for example, if I'm preparing a complete product detail for US, I cannot submit that to EU. Again, I'll have to prepare, reformat it, restructure it, and then I'll have to submit it to EU. Then if Japan needs, or Southeast Asia needs, or any other GCC country needs to be filed, then I'll have to again restructure it, reformat it. So this is a high workload, and repetitive documentation is involved, increased cost, of course, and there is a high probability of inconsistency with the documentation. So there is no common terminology mentioned for each sections of the module and all that. So to avoid this ICH Council has formed, and after the ICH Council or regulation came up, depending on the ICH guidelines, they prepared the documents and submitted so that every country has a common format, common structure, and it is of the job. So in ICH, there are 4 different guidelines as safety, efficacy, quality, and multidisciplinary. Out of which multidisciplinary guideline set is there. From that, ICH mod M4 is the guideline which we follow for the CTD preparation. So in that, ICH M4, based on ICH M4, we have, this is the CTD overview, just a pyramid. So in this pyramid, there are 5, you can see the 5 modules. Module 1, regional administrative information. So this module 1 is not common to all the countries. It varies because there we will give the administrative information, and they will have certain set of requirements. Like some people will require risk assessment study. Some countries may not require that risk assessment study, and different, it varies. About the product I am talking, okay? So to submit a product dossier, first of all, you need to have an animal study. Why we are doing animal study? We will be understanding the safety of the product before trying in the human beings. Then we will have an efficacy trial, which we carried out in humans. After that, what are the quality aspect we follow, maintain during the manufacturing and the quality control? All this information will be there. So all this information put together, we will compile the CTD, and that needs to be submitted. So all this information we will give into these 5 modules. Module 1 for the regional and administrative information. Module 2, CTD summary will be there. Module 3, quality, that is called CMC. Chemistry, manufacturing, and control. Chemistry means R&D development. Manufacturing is the production related document. Control means quality division will check the quality of the product, purity of the product, and that details. And then comes module 4. So in module 4, we will give all the animal trials that we have done to ensure the safety of the product. In detail, we will see each module in later slides. Module 5 will have the clinical study reports. So in each slide, for example, even though there are 5 different sessions, each module has its own purpose, but they all are connected logically. There is a logical connection between each module. So next slide. So first, module 1. For example, today I am my product, I want to register it in US. So for that reason, there will be a module 1 format for US, specifically for US. that module 1 format will not be working for Japan, China, or uh, uh, any other GCC countries or Asian countries. It won't work because the application form structure will be different for each country. For US 356H or 1571F, different forms, application forms are there depending on your application. So EMA or MHRA or EMA of UK and EU, that varies. So this particular module is the one module which is unique to each region and it is not harmonized globally. Out of the CTD, this module 1 is not harmonized globally because it has a different region specific requirement. So application form will be there, product information, for example, labeling. In China, we will have to label the product in Chinese language along with English, and summary of product characteristics, SMPC. Summary of product characteristics we need to give, patient information leaflet, how to use, when to use, what is the dose, what dosage form, all these details will be there in this. So other than that, our GMP certificate, whether our facility is GMP facility, and if that is a GMP facility, we will be having a GMP certificate, and regulatory correspondence with other countries, and what are the previous regulatory approvals that we have, and then risk assessment for during the manufacturing of this product. Each stage, what could be potential risk, what are the risks associated, and what are the mitigation strategies that we have in place so that we will avoid the risk and produce a successful product with a purity, quality, efficacy as we claim. So all this will be in the risk assessment document. So in the module 1, we will be including all these documents as per the requirement of each countries. That is about the module 1. When we go to module 2, this module 2 is a summary of what is there in the module 3, module 4, and module 5. See, CTD document is a huge data, which will have your company's profile, who is the manufacturer, what is your facility, what is the facility layout, details, everything. And then what are the quality control systems that you have in place, what is the manufacturing procedure you have, what is the quality control systems, or what are the quality tests you do, then what are the clinical data, non-clinical data, everything will be there. So for the regulator to see this module as such, they will not get an overview. So before getting into each module of 3, 4, or 5, they will go through the module 2. They will just see whether each section of the module 3, 4, and 5 has been filled, or have you given the complete data set required for that particular subdivision or subsections of module 3, 4, and 5. Then if this module 2 gives complete clarity, it should be very crisp and clear data. Your module 2 should be, because you are giving an overview of what is there in your module 3, 4, and 5. So here, as I said before, module 3 will be of quality overall summary, 4 is of non-clinical and clinical overviews, and non-clinical and clinical summaries. What are the clinical trials you have undertaken? What are the non-clinical trials you have carried out? All these details in a summary form, very crisp and clear, short, brief, you will be giving in the module 2. Okay. Now comes the most important section, which is CMC, Chemistry, Manufacturing, and Control. Here in this module 3, mainly there are 2 sections. Drugs, that is called drug substance, and drug product section. See, whenever you manufacture a drug product, okay, so for example, biological product we will take, for example, a monoclonal antibody, bevacizumab. This is I'm just telling for an example, bevacizumab is a monoclonal antibody which is used for the treatment of cancer. So for this product, you need to give the CMC section. You need to fill the CMC section. For the CMC section, there is a section called, okay, somebody had raised a hand. Actually, I cannot see who it is.
Rishma, what we will do, we will take the questions towards the end.
Okay. So there will be a module 3 that will have mainly 4 sections, but out of which drug substance section and drug product section are the most important part. In this, as I said, bevacizumab, that is a monoclonal antibody. So there will be 2 sections. So in the drug, that is starts from 3.2 S.1. Here in ICH, as per ICH guideline, you will keep a section for the drug substance. It is called general information. In drug substance section, that 3.2 S.1 is called general information. In this section, you will give the general information about the drug substance that you are producing. So in case of bevacizumab, there will first you will go for the fermentation of your cloned cell. After that you will produce the API or the bulk drug substance that will contain in the drug substance section. Once you have a bulk drug, then you will do the formulation. You will pack it, label it, and make it into a vial form or the injectable, that final form of your medicine. That is called the drug product section. So how the drug substance has been developed, what are the process involved, that you will give in the drug substance section. So each section has this number. This is very important. For example, 3.2 S.1 of drug substance section is called general information. Whether the CTD you are submitting in India, for example, not India, in US, EU, Japan, or Saudi Arabia. So this sections of CTD will not change. It will be 3.2 S.1. S means drug substance, that substance. That is drug substance section. 3 means module 3. Module 3 is 3.1 will be overall summary, table of content. Then 3.2 S.1 will be general information. You will give the general information under the general information section. For example, what is the nomenclature, name of the product. There will be, like, for example, I told bevacizumab, that is a generic name of that product, and manufacturer will have a brand name. So these details you will give. Then comes the structure of what is the structure of that product. I mean, in a gist you will give. Then what are the general properties of this product? All this you will give in the general information section of drug substance. And drug substance, when it comes to manufacturing section, that is 3.2 S.2. I will, at the end of the session, I can show you the module 3, and I'll show you and I'll explain you if you need in much detail. There in this section, S.2 section, second section, it is about manufacture. For example, first you will have to give who is the manufacturer. For example, my company, Sansame Biologics, and my company's address, details, what is the location, everything I'll give it in the manufacturer's details. After that, you will give the description of the manufacturing process, how you have manufactured that process from starting of the drug substance only. Drug product, there is a separate section. Only you will explain about the drug substance, how you have manufactured. Okay, starting from the cell culturing, and then you have gone for the fermentation. After that, viral activation or chromatography. Then after that, you will go for the upstream will be there, downstream process will be there for the manufacturing. So your upstream process you will explain, downstream process you will explain. After that, you purified bulk drug will be there. All that we will be mentioning in the manufacturer section, S.2. You will give the idea of how you manufacture. Then comes S.3. Third section is characterization. So, before characterization, in manufacturing, you will give some more details. What are the raw materials that you have used? What are the control mechanisms that you had? Okay, all these subsections are there. Under manufacturing section, there are around 6 subsections are there. So each section is like, first you will give the name and details of the manufacturer. Then you will give the details of the description of manufacturing process. Then you will explain how the materials, the raw materials are controlled. avoid contamination, because you need a pure sterile product. So what are the systems in place for you to maintain the sterility of your raw material? So that is called control of materials. Under that, you will give control of materials. After that, what are the critical steps involved in the manufacturing procedure? So, for example, if your product is pH condition, for example, that pH should be of 6.3 to 6.6. Other than temperature conditions, it should not be more than 37 degrees Celsius. Or cell viability, it should be of 2 million cells per ml. Like that, different control parameters will be there. So you will give the details of what are the critical steps involved in your manufacturing process. Then process validation. Okay, now you have a process in place. After your R&D, the R&D team might have developed a process to prepare this product. So this product, after following this process, you are getting a consistently qualified product which has the same purity, same efficacy, same quality. If it is there or not, you should have a validated process. Then only you will get a quality product consistently over the period of this manufacturing. There will be evolution in the process. Those and all, like some details are involved, changes are there, that in later sessions we can discuss. But so these details you will mention in the manufacture session itself. So that will give a clear idea about how the product has been manufactured. And the third section is called characterization. So characterization, for example, this bevacizumab, it's a protein. So how the structure of if you're a biotech student, or you might be knowing, there is a primary structure, secondary structure, tertiary structure and all. Protein, how the protein will form, that central dogma, DNA to RNA, mRNA, then the protein will be forming. And after protein formation, post-transcriptional modifications will be there. So all these details, how the product has a structure, how many amino acids. For example, in bevacizumab, there are 1339 amino acids are there. So if there is a small protein like brazil or something, they have only like 53 amino acids. So biologic product, they have 150 kilodalton big molecule. But a small molecule like 6 kilodalton small molecules are also there. So these details, characterization, you will do glycan profiling, peptide mapping, all this. I don't want to confuse you for the first time about the product and all. So this characterization, you might be knowing SDS page, for example. This is a small part. So in SDS page, your molecular size separation you will get, right? Picture when you do the silver staining. Usually for the regulatory submission, we will not use the methylene blue staining and all. Silver stained SDS page picture you might be knowing. So in that you will get the bands of this product. That will give idea of your molecular size of the product. So these kind of details you will give it in the characterization of the drug substance section. Okay. And next section is S.4, that is control of drug substance. Okay. Here till now, first general information we'll write. Manufacturing section is from the production team. They are the manufacturing department. They will have their set of documents, BMR, process validation, all these documents they will be maintaining, the production team of the pharma company, and they will give the details. Okay. And the characterization part, this part R&D might have done. They will do the characterization, but still after the production, again we will check our manufactured product, whether they are complying to the structure what we wanted or not. That we will check. Other section, next section is control of drug substance. So once your drug substance is ready, after the manufacturing product, you have the bulk product in hand. Then you want to test few parameters depending on, for example, as I told you, product has manufactured, but is it of your required quality or not? You want to test it. If that is of low quality, then we cannot give it, because that will create further issues, adverse events in the market. When it goes to patients will have problems with this product. So for example, you are claiming that it's a sterile product. Sterile product means there should not be any microbial organisms present in that. So you will check the pure sterility of the product. It should not have any bacterial endotoxins present during the manufacturing. See, manufacturing unit will be a big facility. Many people will be involved. Many machineries are involved. But you will be maintaining a GMP facility, good manufacturing practice, so that all these sterilities, impurities, contaminants and impurities will not be introduced to this product. Why? Because you will maintain certain set of rules, some SOPs to avoid these conditions. So how the product looks like, appearance you will check, pH you will check. Again, the glycan profile. What are the impurities present in this? You will check. Impurities means, for example, Beva, it's a biologic product, right? You will be using a cell culture. You will be growing it in the CHO cell line. So that cell, other than this is a protein, you just need this protein. Other than this, whatever is there, it's an impurity. You don't need that. So that is the reason you will have certain 2-3 chromatographic techniques you will use to filter it and remove this other impurities. Viral impurity, you will filter it to remove the virus, everything. So certain set of test parameters you will mention in this specification. As per the test parameters, what shall be your result or a limit of result, that criteria would be mentioned. And after testing, you should check whether that particular parameter is meeting or not. For example, the color of the product should be transparent. It should not be opaque. That liquid should be clear, transparent liquid. If it is opaque or yellowish or greenish, then you cannot say that it is out of specification. Okay, so you have given a criteria. Within that criteria, it should remain. That is called the specification, how the drug control is maintained. So for this specification is also, there is a certain set of ICH guideline, which will ICH Q6, I think, that will give the idea of how your specification limits to be kept, what are the parameters, how to present it and all that. Then comes reference standards. So you will see bevacizumab you are manufacturing. In market, there are different kind of different manufacturers who are already producing bevacizumab. So you have to test for this product, particular drug substance. Not only this is called reference listed drug, the reference product in the market with other companies. For example, company X has a bevacizumab, company Y have a bevacizumab, and I am company Z. I am manufacturing the beva. So my bevacizumab should have equal quality of the X and Y, or it should be better. Okay, it shouldn't be lower than that existing product. The quality should not be lower than the existing product. And so some products, this market available product reference product will not be there. And some product, for example, another biologic product, albumin and IVIG. IVIG is a biologic product. You all know immunoglobulin G. So this immunoglobulin G is extracted from the human blood. As we are taking it, so we will collect different human plasma, and we will process it and extract the immunoglobulin. And that after the purification and all, we will vial it, and final formulation will be IVIG. So for this product, there is no biosimilar product is not, we cannot consider. Why? Because we are collecting plasma from different people, and each plasma will have a different profile. So we cannot call it as a biosimilar product. It is a biologic product. So we will not have this product we cannot compare it with the market existing product. So then what we do is, EDQM. I mean, there are other regulatory, lab regulatory authority approved labs, which will give the reference standards. That standards, for example, for albumin or IVIG, there should be certain specification parameters to be passed. You should test that parameters, and then that should be within the limit. Then only you can release the product, right? So this reference products will be available. So this reference standards, along with the reference standards, you will compare your product, and that comparison, if it complies, then you will move further. So this reference standards details from where you procured, how you are maintaining, diluting it, or once it is, every time you will not be able to use the reference standards procured from outside because it's very expensive. So you will dilute, and in-house standards you will prepare. So how you have prepared that in-house standards, all these details you will be mentioning in the reference standards section. Then comes container closure system. Okay, so the bulk product you have manufactured, if it is a biologic product like bevacizumab and all, you will be storing it in big plastic bags. That plastic bags, you cannot just store it in any plastic bags. These plastic bag also have some specifications. Their primary packing, first you will put it in that first bag or the bottle or the package which is in contact with the drug substance is called primary package. So that bag, LDP bag, with how many layers, how it is, and what are the tubings associated with that bag, every details you will have to give it in the container closure system. What is the bag, and after the first primary packing material, if there is any other outer layer to that primary bag, that is called secondary packing material. And above that, if you have any other cartoon boxes or anything, you will mention that as tertiary packing material. So all these packing materials, from where you have, who is the vendor, from what is the specification, and for example, I told you it's a plastic bag. If it is a plastic bag and you're storing it for a certain period of time, your shelf life period, then if, for example, you want to store it for 2 years or more than 2 years, you are storing it in a plastic bag, right? So there is any chemical which is leached from the bag to the product and changing the quality of the product or not, that you need to understand. So leachability, extractability, all these parameters needs to be tested of that container closure system. And that details will be provided in your 3.2.S.6, that is a drug substance section. Okay, there you will provide. And then comes the another important section, stability. Okay, you have manufactured a product which is of very good quality, which is of very good efficacy, and you are planning to commercialize it. So from my facility, I have to transport it from my facility, and I have to distribute it to the required distributors. And from the distributor, it will go to the patients or hospitals, wherever. So there is a duration involved, transportation duration. After that, it will be there in the hospital shelf for many days or months. And after that, they will prescribe and it will go to the patient. So it may take some time. So whether this drug substance or drug product which you have manufactured has the stability, is it stable for this long duration of transportation and putting it in the during the distribution time and everything, whether that will be stable, and you will this will be exposed to different temperature conditions, humidity conditions, and some drug products will be light sensitive. I mean, if it is exposed to, you might have seen brown bottle drugs also, because it is light sensitive. That is the reason they pack it in the brown bottles. So if it is light sensitive, how much? So we, first of all, we have to set a certain shelf life period for this product. Okay, so this product is stable for 1 year, 2 year, or 3 years. During that time, you will have to put it in the stability chamber. We call it a stability chamber. And for example, your drug is stable at only at 2 to 8 degrees Celsius. Then you have to set the stability chamber at 2 to 8 degrees Celsius. And for that drug will be stored in that for, if you are claiming 2 years, you will store it for 2 years. That is called a long-term stability. There is another stability called accelerated stability. For example, this stress or degradation pattern is there or not. You will check the stress condition also. So in accelerated condition, for example, your drug should be stored at 2 to 8 degrees Celsius and it will be stable for 2 years. So if you're testing accelerated stability, what you will do, your drug, you will keep at 25 degrees Celsius. That is in the ICH guideline of stability. There is ICH Q1, which has explained what shall be the stability condition for different temperature zones. The whole world has divided into different temperature zones. Zone 1, Zone 2, Zone 3, Zone 4, A and B. India and most of the Southeast Asian countries fall under the Zone 4B condition. And there are Europe, US and all is there. They are Zone 2 condition. So depending on your zone, temperature zone, the temperature condition also varies. Okay, so Zone 4B means we if it is a room temperature condition drug, 30 degrees Celsius and 65% RH condition is the stability parameter requirement. So at what temperature condition, whether you have done long-term stability, whether you have done accelerated stability, all these details you will mention in this section S.7, stability section. There you will say, based on our study, this is the report. For example, 3 batches ka stability report you will have to submit. Okay, accelerated and long-term stability. That details and your data, raw data, and if you have any stability commitment, for example, after getting the manufacturing license application, usually every year you will be manufacturing the product, right? At the end of the year, you will be, beginning or end, one batch of the product, you will again keep it for stability testing. So that is as a commitment, you can say that after manufacturing license application, one batch production batch will go for an annual stability testing. That details and all will go in the stability section. Okay, this is all about drug substance. Same way, when it comes to drug product, there is one section extra. Okay, here also same, description and composition. In this description and composition, you will give the information of your drug substance. This drug substance you have used. Along with the drug substance, what are the excipients? Other than in a formulation, other than the API or the drug substance, what are the other components? They are called excipient. So how much quantity of API you have used in, for example, 100 ml vial? How much is the API? What are the other components? Excipients, like for example, water is also an excipient. How much quantity of water is there? That details you will give in the description section. Then comes the pharmaceutical development section. Here, PDR, product development record, you will submit usually. This pharmaceutical development, how you have gone through the development. In detail, drug substance development you have given in the manufacturing section, right? Same way, here it is called pharmaceutical development section. This section, your drug product details you will give, the formulation, how the formulation, excipients and API combinations you have prepared. Overages. Overages means, for example, there will be some extra of this API or which will be added so that future degradation can be, sorted. So in detail, I'll not go to that. Those are all confusing part. So this overages, physical and biological properties of this drug product, all these details we will be producing in the pharmaceutical development section. And again comes the manufacture section. What are the process, how you have gone, and what are the controls in the drug product manufacturing. From the bulk only you will process your drug product. So what are the process? What are the process controls? And then excipients. Here in drug substance, that excipient section was not there. For the drug product, that excipient is there. So here, along with the drug substance, what are the other components that you have used? That particular component ka specification. For example, water is an excipient, considered as an excipient. Water, what is the purity of the water? What is the pH condition of the water? Uh, um, whether there is any microbial contamination is there, heavy metal contamination is there. All these test specification will be there for that excipient. Same like water, there are other components. So all other components, there will be set of testing parameters and that limits and what is your result. All this will be mentioned in certificate of analysis. Those documents and details will be given in the control of excipient section of drug product. And after that, control of drug product section. Same like drug substance specification, drug product also will have a set of specification criteria. All that detail will be given in the 3.2.p.5. This, if you say 3.2.p.5 section, which section it is? You ask an American drug controller or a US, European drug controller, they will say that it is control of drug product, because as per the ICH M4Q, they have, you shall give these details at this particular section. So these details will be provided. And then reference standard, same like drug substance, which will have a reference standard. You will have a drug product reference standard. And this also will have a container closure system. Instead of big bags, you will have 100 ml vial, 50 ml vial, 10 ml vial, and that bottle, and that will have a rubber cap. So specification of the rubber cap, the glass bottle, everything, that is the primary container closure system. After that, there will be a cardboard box type, GSM box will be there. So how much GSM, 350 GSM, 400 GSM, what is the GSM of that product? That is about the secondary packing material. So there are different GSM box you will be packing in a carton, 5-ply shipper or 3-ply shipper. These are the specification of the shipper. All these details will go into the container closure system of drug product, 3.2.p.7. And then comes the stability. So the final packed drug product. For example, as I said, there will be a glass bottle with a rubber tube, and then aluminum covering will be there on the top, and that will be kept in a GSM box. And that box needs to be kept in the stability chamber, and that stability chamber conditions for how long you have kept. In between, for example, first 1 year, every 3 months, you will be checking whether the product is stable. There will be a set of specification which is passing or not. All this will be checked in your stability. And same like drug substance stability, we will give all the details, commitment, everything in the drug stability section. So for this ICH, for this module 3 quality CMC section, the guideline which we follow is ICH M4Q. Q means quality. So ICH M4Q guideline is there. As per that guideline, it is very clearly mentioned what should be given for a new chemical entity, which means a drug, a new biological entity or a biotechnology product. So that is clearly mentioned in the module 3 guideline, which is ICH M4Q guideline. I'll show you at the end of the session if there is time. Okay, based on that, we are preparing this document. Okay, now comes to module 4, non-clinical study reports. So non-clinical study, you will be carrying out these non-clinical studies in rodents or non-rodents or chimpanzees, or depending on the drug product or biological product you are developing. Depending on that, you will be selecting your animal, lab animal. So there you will conduct different pharmacology studies, pharmacokinetic studies, toxicology studies. All this will be done. For example, pharmacology study means, okay, how the drug works inside the body. So method of action of this drug inside the animals is pharmacology study. Okay, so its intent is how the biological effect of this drug inside the animals, or how it creates adverse events, or which organ it is targeting. All this you will get to know. I mean, it's not accurate, but approximate idea you will get from this pharmacology study. So first you will conduct a non-clinical trial in the animals. And nowadays, that also I should mention, nowadays many countries, they are avoiding this animal trials. Instead of that, they do in vitro trials in cell lines and all, they will do, so that cruelty on animals can be reduced. Okay, so how this drug works on the animals, or mode of action of this drug in its body, or off targets. For example, you are developing a drug for liver. It should function, it should go to the liver, and receptor, it should attach to the liver receptor. Whether it is, instead of going to the liver, whether it is going to the lungs, kidney, or any other part, or any other bloodline, or how it is, how its mode of action, that you will understand from the pharmacological evaluation. And then comes pharmacokinetic study. So in this pharmacokinetic study, you will, after taking the that product, how it gets absorbed, how it gets distributed, how it gets metabolized in the body, and how it gets excreted from the body. All these details will be studied in the pharmacokinetics section. Okay. And next is this toxicology study. Okay. So you have developed a product. How much dose you should given to you should be given to the human, that you need to understand. If that dose is toxic, has a toxic level, or is it sufficient or moderate or low dosage, this you need to understand. For that reason, they will do acute toxicity or repeated toxicity. Acute toxicity means a higher dose of the medicine will be given to the animals. Repeated dose toxicity means for a repeated 28 days, 14 to 28 days, they'll repeatedly give a small dose of this medicine to the animals and will check the changes. And then they will check the carcinogenicity or reproduction, if there is any adverse events for the reproduction or related this thing. And then genotoxicity, whether it is interacting with the genes and causing any toxicity or cancer causing. All this kind of toxicity studies will be conducted in the animals before performing a human clinical trial. All this data will be mentioned in the module 4. And all animal testing data, entire data will go here, and this will help us to establish the product's safety before conducting a human trial. And for this part, we will use the guideline ICH M4S. S means safety. This is a safety trial. So what are the testing parameters, how to represent, everything is mentioned in the ICH M4S guideline. Depending on that, module 4 will be prepared. Okay. Next comes module 5, the clinical study report. So here, clinical trials, as you all know, there are different stages of clinical trials: phase 1, phase 2, phase 3, phase 4. Studies will be there. This phase 1 also, phase 1, 2, 1A, 1B, 2A, 2B, like that. So all these trials, clinical trial data, your in previous safety and efficacy summaries, studies details, and case report, this will be conducted in humans. So all human trial data, everything will be compiled, and case report forms of each subjects or patients who have been in the study will be comprised and given in the module 5. So for module 5 will give you, proves the benefit risk profile of the product in humans. For example, as I told you before, whenever you develop a product, it should have some benefits, and all drug will have a side effect. Okay, so the side, for example, a cancer patient, they'll be in the terminal stage or initial stage. For them, curing the cancer or relief from the cancer is the benefit. There will be some side effects. If that risk is lower when compared to the benefit, if that is more important, then the drug is good to go. You can proceed with the development and it can go to the patient. So that this, after the clinical trial in humans, you will get to know whether this product has a benefit risk profile which is advantageous or not. Okay, that all these details will go in the module 5. So this is the complete dossier: module 1, module 2, module 3, module 4, module 5. This comprises the dossier as per CTD, that is a CTD as per ICH guideline. And the guideline which I told you, ICH M4 guidelines is M4, Q is for quality session, S is for non-clinical trials will be gone into the S section, and efficacy ICH M4E will have the clinical trial details. So other than that, there is one more guideline, ICH M48, which will have the electronic common technical document. Okay, so your dossier you can submit in two formats. One is electronic, one is non-electronic, PDF formats or paper. In India, till recently, there were paper submissions were also going on. But now they have made it completely electronic. So in electronic submissions, you can provide all this PDF files. You can file it, put it in a floppy disk or a pen. Nowadays, pen drive. Initially, it was disk. You have to put all the PDF files with the separations, numbers, and all. You will have to submit. But now there is a electronic common technical data requirement. US, EU, US, UK, and all, Japan, and many Southeast Asian countries, they require only dossier only in ECTD format. So there will be a XML backbone. I mean, like, you will have the documents, but there will be hyperlinking. That is entirely a different lecture is required for that. So that's the reason I'm not explaining. It's already, I think it's already 1 hour. Okay, so there is this ECTD submission is very important. There are certain softwares which can help to submit the dossiers. So when you have a ECTD, so this is same, sections are same. Drug substance section will be there, drug product section will be there, appendix section will be there, region specific section will be there. Every section are same. So these sections, but it will be in an electronic format. Everything is just a click away. So whatever we have submitted, that will go to the regulator. Regulator will see whatever the corrections or the queries they raise, that will be communicated electronically. We can see anytime, and what are the query responses that we have made. Everything electronically managed, and it is easy to handle compared to the simple CTD or non-electronic. This is very easy, and each versions, for example, after the submission, there will be queries, then you will be updating and amending this, and CTD versions will change, and all this version we will have a control here. But in the other section, even though we can maintain different folders, files needs to be maintained, but in this there will be an XML backbone, so electronically you can navigate. And most regulatory agencies nowadays, they mandate that you should submit your dossier in a ECTD format. So CTD accepted by, as I told you, FDA, US FDA, EU, EMA, PMDA, Japan, Health Canada, WHO. These regulatory agencies, they prefer CTD format as per ICH M4, Q, E, S, 8. These guidelines, based on these guidelines, they need documents. So with this, you will give a single dossier for whichever global country or whichever country around the globe you are submitting. Most of the, yes, 95% of the time, your single dossier will go. But there are different versions of CTDs as well. That and all we will see in different classes, because Asian countries will have just a slight change. instead of module 2, they will not have a module 2 for the summary. They will directly have module 3, 4, 5, like 1, 3, 4, 5. Okay, so only 4 modules will be there. So the CTD is globally accepted. Only one module changes regionally, because they have region specific requirement in the module 1, so that is not harmonized. Okay, so there is few tips. After giving the class also, you might, if you are a fresher or you might not have done this, you still won't have that idea. So you can start with ICH M4 guidelines, like ICH M4Q, M4S or M4E. You can see, I have it in my folder. You can, if possible, I'll show you. Otherwise, you can see your check with ICH, type ICH guidelines. You will get all the ICH guidelines, safety, efficacy, multidisciplinary, quality, everything you will get. In that multidisciplinary section, the CTD guidelines will fall. So what you have to understand is, you have given a about drug product in module 2, you have given certain set of details, but in the detailed section in module 3, drug substance section, you have contradicted it, or you haven't consistently given the data, then that will be a problem. So you should have that consistency maintained throughout the documentation. And your module 2 should be concise and clear. Everything you need to summarize. So first, you will not prepare the module 2. You will prepare your module 3, module 4, module 5. After completing your all modules, you will come back to module 2 and you will prepare your module, because you have to summarize everything in that. So consistency and clarity, you have to give clear data of what you have to be done. That needs to be given in your modules, CTD. Okay, that's all for the session. Couple of questions I have, but before that, I just want to understand what you guys got from this discussion. So back to you, Sujit. You can navigate it further.
Thank you, Dr. Reshma. We will take 1 or 2 questions. I guess Sandeep Suresh has raised a hand. Yes, Sandeep, you are allowed to talk. Sandeep?
Okay, there is one question.
I am audible?
Yeah, okay. Yeah, yes, it's audible.
Yeah, my question I just posted just now, actually. The role of AI in ECTD submissions in coming future, and will be the queries which will be raised by the regulatory inspectors will be reduced, and the approvals will be very quick. What is your opinion on artificial intelligence in coming days?
Artificial intelligence, see, definitely that is going to ease off your work. Nowadays, even Perplexity or ChatGPT also can give, if you are giving a drug product requirement, what is the required for this particular product to be. For example, resin is a protein, I mentioned you, right? So what is the regulatory stand of this product in different countries? If you give, they will give you the idea, and what are the documents needs to be submitted. All this, when you prepare the strategy, that part, I felt that it is very helpful for me, because I've been using this AI like that, I mean, in a general way to understand. But when it comes to regulatory submissions and all, that needs to be more regulated, because CTD is all about confidentiality, confidential document of the product and the company. So that way, I don't know how much in my experience, we haven't used it yet. Only for the initial strategizing and strategic preparations we have used. But other than for this submission part and all, we haven't yet used it. But it is going to be a very good hand supporting for us to, navigate through. And the regulator, I think definitely the submission and everything will be faster.
Okay, thank you.
Dr. Reshma, we can take one more question.
Somebody has asked, it is very informative session so far, and during generics dossier filing, what will be mentioned in module 4 and 5, and we don't do any non-clinical. Yes. So when it is a generic product, yeah, I was just saying in a biological aspect, if it is a biosimilar generic product, nowadays even generic product, they ask the non-clinical clinical trials. There what you will fill is already existed studies, research articles would be there, or review of literatures, you will say. Abstracts of those studies you will mention with the abstracts and the corresponding author, the first author, second author's detail, and the abstract. In each section we will give non-clinical and clinical trial. What are the previous studies has been conducted and why you are claiming that it will be effective or it will be safe. So each section based on this ICH, M4S and M4E guideline, based on that you will prepare the summary, right? There abstracts you will be giving. And that's what we have done. Yeah.
Okay, we will come to that one last question, Reshma. That a student since we have answered 2 industry questions, we will answer one in student question also. I am a biotech student. You can see that question. Second last question. I'm from Pavani. I am a biotech postgraduate. Can I enter into regulatory phase career, or only pharma are eligible, madam? Career growth.
No. See, I have completed my graduation in microbiology, post-graduation in microbiology, and PhD in biotechnology. I am not from a pharma background person. So, see, nowadays there are many pharma companies. They are coming with the biotechnology products. So they need biotechnology people. And there are a lot of opportunities for you guys. Initially, people used to think that biotechnology, I have taken, there's no scope. It's not like that. Pharma company, there are many pharma companies, they are seeking freshers for with biotechnology knowledge, because this rDNA technology and all, vector preparation, cell culturing and all, you know better than the pharma. Your DNA proteins in this DNA central dogma and all, nobody has to explain it to you. You already know it, right? So all this needs to be comprised in the modules. So you are eligible, highly eligible. Don't worry about that.
Okay. So what we will do, there are more questions. We will note down these questions and try to answer them because we have exceeded the time. We'll stick to the time schedule. I'll pass on this question to Dr. Reshma, and I'll ensure that you get the answers. Is it fine?
Sure.
I am really thankful to each one of you for attending in large numbers. Next Saturday, we have scheduled for molecular docking. It's another important topic which is coming up, especially with the Indian pharma, Indian CDSCO also looking to make it compulsory with respect to the regulatory aspect. So we'll come to molecular docking in the next session. And of course, you can also pass on your reviews, comments. We have already pasted the review section. If you want to know anything about any particular aspect of pharma, please feel free to post that particular question in the group, not in the group, in the review section. I will ensure that we come up with a topic which is suitable for that. Okay, thank you, Dr. Reshma. It was a really wonderful session, and we all would have learned a thing or two which we were not knowing with respect to CTDs. It's a never-ending topic. We'll try to get more topics from you as per your time schedule, because in the sections in the chat session also I can see a lot of questions related to which were not explained by you. So I guess we will have another session from your side also. Thank you so much for coming on this weekend. Thank you, everyone, once again, and happy weekend. So we'll conclude the session, and as I said, we will be posting this recording in the YouTube channel also. You can hear it again, and if you have any questions, we'll be there to address them. Thank you so much. Bye, everyone.